Hello everyone, welcome to my channel Nurturing Intellect. Hope you all are doing well. In our today's class, under the chapter Biotechnology Principles and Processes, we are going to study polymerase chain reaction. So let's begin. Polymerase chain reaction consists of three words: polymerase, chain and reaction, which means reaction means it's a chemical reaction. Polymerase is the enzyme DNA polymerase, and chain means it consists of series of reactions which are repeated one after the other. So in this reaction, the DNA polymerase, as we know, it replicates the DNA. So in one cycle, after completion of first cycle, we get 2 DNA molecules. After completion of second cycle, we get 4 DNA molecules. After completion of third cycle, we get 8 DNA molecules and so on. So you will find that after completing few hundred PCR cycle, you will obtain nearly 1 billion copies of the desired DNA. So PCR is the short form of polymerase chain reaction. In this, we are amplifying the piece of DNA and generate several billion copies of the DNA. Now, a question must be coming to your mind: why to make so many copies of DNA? Why there is a need? So, first thing, it is because it helps in diagnosing. It is very useful in diagnosing infection and infectious diseases at an early stage. For example, if a patient is suspected of HIV infection at an early stage, because the viral concentration is very low, we won't be able to detect the virus. So what is done? We collect the blood from the patient and then we subject it to PCR. If the virus is present, the DNA of the virus will get amplified, and when you test it with the proper technique, you will be able to identify whether the person is HIV positive or not. It also helps in identifying the type of virus and to study the properties of virus. Besides this, it has several other usage, like in crime investigation, for genetic research, molecular biology, and recombinant DNA technology. In this video, we will be focusing on recombinant DNA technology. PCR is carried out in a machine which is known as Thermocycler. So, the name itself suggests thermo means related with temperature, cycler means it is repeating or varying the temperature. So in this, there is fixed program where we can vary the temperature and we can amplify the gene of interest to many times or multiple times. So there are PCR tubes in which we add the various components which are needed to conduct PCR. So let's see what components we need. First is DNA template. So it is the segment of DNA which we need to amplify. That is our target sequence. Second, we need an enzyme which is going to replicate this DNA. The name of this enzyme is Taq polymerase. The name Taq polymerase is given because it is isolated from a thermophilic bacteria, Thermus aquaticus. This bacteria is a heat-tolerant bacteria. So, the enzyme so obtained is able to tolerate high temperature, which is carried out in the PCR. Other enzymes, they get denatured at this high temperature. Then we need DNTPs, that is deoxyribonucleotide triphosphates, as the raw materials which are used by Taq polymerase to synthesize the DNA. we need a buffer solution in which we add magnesium chloride, where magnesium is acting as a cofactor for enzyme Taq polymerase. It activates enzyme Taq polymerase. We need a pair of primers, and these all the components, they are taken in the PCR tubes altogether. Place these tubes in the thermocycler and carry the PCR reaction multiple times. Now, what are primers? Primers, they are small fragments of DNA which contain 18 to 24 nucleotides. Okay, so here you can see this is a primer. So it is 18 to 24 nucleotide long, and these primers, why we are using DNA primers? When the replication is taking place inside the body of any living organism, RNA primers are used. But whenever we carry any reaction outside, that is in vitro, we make use of DNA primers because they are more stable at high temperature. Now, addition of primer is important because the DNA polymerase, in order to carry out polymerization, they need a free 3-hydroxyl end. So, they bind with the 3-hydroxyl end of the primer and then carries out the polymerization of DNA chain. The primers which are used in PCR, they are in pair and they are called forward primer and reverse primer. Forward primer is the one which binds with the template strand of DNA. Template strand is the strand of DNA which gets transcribed. And reverse primer is the primer which binds with the coding strand of the DNA. So, in PCR, we make use of pair of primers, forward and reverse primers. Now, let's see the steps of PCR. PCR involves 3 steps. First is denaturation, second is annealing, and third is extension. Let's study them in detail. So, first step, that is denaturation, it is carried out at a very high temperature, that is 92 to 96 degrees Celsius. At this high temperature, the hydrogen bonds present between the two DNA, present between the complementary strands of DNA molecule, they break, and thus the two strands, they get separated. So that is important, then only the polymerization can be carried out. After denaturation, so at the end of denaturation, we are getting 2 strands of DNA. Then we lower the temperature to 50 to 55 degrees Celsius. At this temperature, the primers present in the reaction mixture, they bind with the template DNA strands. And this step is known as annealing. The third step is extension. Again, after 45 seconds, we are increasing the temperature to 72 degrees Celsius. Taq polymerase is most active at this temperature. So, the Taq polymerase, it binds with the 3-hydroxyl group of the primer and start adding DNTPs to the new DNA chain. And in this way carries out extension of the DNA chain. So, at the end of one cycle, we are getting 2 DNA molecules. So, this cycle repeats several times, and after about 3 to 4 hours, you will get nearly 1 billion copies of the DNA. So, what products we are getting? We are getting the amplified DNA fragment. This fragment is amplified by the Taq DNA polymerase. So, these polymerases in the beginning of the chain extension, they add adenine group to the amplified DNA. Now, to this we add the cloning vector. So, we can then by adding ligase, we will get recombinant DNA. So we will get several copies of recombinant DNA. If we have amplified the gene of interest 1 billion time and vector DNA also 1 billion time, so we will get 1 billion, nearly 1 billion copies of recombinant DNA, which can be used to transform nearly 1 billion bacterial cells. And these bacterial cells, they can be cultured on large scale. So this is how the PCR is useful in recombinant DNA technology. The gene of interest can also be used directly by the method microinjection and bioelastic gene gun method to introduce them into the plant or animal cell. So this is all about PCR. In our next class, we will study about the different steps involved in recombinant DNA technology. So, hope you must have liked this video. If you liked the video, please hit the like button, comment and subscribe to my channel. Don't forget to hit the bell icon so that you will get updated about my latest videos. Thank you for watching.